before I start, I would like to go back to the future. So when I started my postdoc more than 20 years ago, if ever I had this kind of tool, I'm sure I would have much more publication than now. The third and final point, everything I'm going to show you are generating biology hypotheses, right? But QIAGEN has been kindly enough, Frank and people before, to allow me to continue what I've been doing for 35 years, publishing papers. That's what I do. So I'm not a bioinformatician. I'm just doing in silico research and collaborating with people at Mount Sinai, NCI, CCF, you name it. And the only goal that the value of this tool bring is when I can publish with them. And they are very happy. And we continue to do that. So that's my goal, and I'm very happy to do that. So for this particular data set, it's about innate anti-PD-1 resistance. You know, anti-PD-1 is a treatment of value for melanoma, for instance. But some of the people are not responding well. In fact, they have what we call innate resistance to it. And the data that Joe and myself have presented this morning was talking about that. And here, I just bring some of the conclusion. It's easier. I'm going to talk about a potential transcriptional program that we have detected. Can be tested in the lab, can be evaluated. We have found what we call a master regulator. This one, if you inhibit it, you can revert potentially that innate resistance to anti-PD-1. So you can test that in the lab. We have found specific isoforms, splicing variants that are of high interest because they are connected to specific biological processes, or they are enriched in certain tissues. And this is one of the power of OmicSoft I'm going to talk about today. We also find a common regulator cytokine signatures that include cytokines and growth factors. And we could do that because we can go and scan all these pre-computed data sets from OmicLand. And this is extremely powerful because now, at my hand, I have over 6,000 data sets I can scan, which was never possible before. And also, because we have that strong connection between IPA and OncoLand, I can go back and select one gene of interest and see if it's connected and associated with another gene. And I can do that in OncoLand. Thank you, Joe, by the way, for that. So Frank talked about that. This is really, really powerful. And literally, I've been paid to play with all these tools together. So today, I'm going to play with OmicSoft, and I'm going to play with IPA and go back and forth. And you will see how that sample to insight is really powerful. So I'm going to really pinpoint two slides. We have processed the data in RStudio. We have found differentially expressed genes and transcripts. We send that to IPA. Then we are going to go in that interpretation of the whole transcriptome. And this is where the integration of IPA and OmicSoft is really powerful. So you can detect pathways and genes of interest, fine. But now we are going to use that IsoProfiler-GTEx, the GTEx data that has been processed by OmicSoft at the transcript level, and will help us to detect isoforms that are enriched in specific tissues and could be of importance for biomarker. We are going to generate novel regulator networks, which is what we call a Causal network. It's a strong feature in IPA. We are going to compare that analysis of the anti-PD-1 resistance across a repository of processed data set from OmicLand. And we are going to visualize that gene, one gene of interest, FGF2, in OncoLand. This is the paper about. They were looking at anti-PD-1 on a biopsy pre-treated melanoma from advanced melanoma patients. Some could respond, some could not. Let's try to investigate what's going on. I like that slide because it means everything to us. You have the OmicSoft, you have the FASTQ data, you have the sample metadata Joe has talked about this morning, extremely important. And you are going to use that power of the lands and expression data into IPA and probably go back and forth. So this is the complete integration. The data has been processed. Joe has mentioned that. And we are already here. This is the window we are going to send the data to IPA. We can set up a couple of parameters directly. I don't even have to make the cutoff in IPA. It's done already here. So I can do the cutoff, the fold change, the p-value, the group max, anything. And you push. submit. And I'm not going to talk about that. I talked about that this morning. And we are going to answer these questions. So remember, the data I sent to IPA will take literally 2 minutes. IPA open, and I'm going to look at underlying transcriptional program today. I'm going to look at other pathways and networks using a simulator called a molecule activity predictor. I'm going to look at specific splicing of interest that are enriched or tied to biological processes. I'm going to bring a strong hypothesis that I encourage to test in the lab. And I'm going to compare that analysis in pre-computed land dataset using the new feature called analysis match that will be released at the end of September. So it's not even there right now. This is the summary of the core analysis. This morning I talked about it's a way of summarize everything. You have top canonical pathways based on overlap, top regulator, top master regulator. So that's simple. This is study at the transcript level for change of 2, p-value over 5, and max count of 10 counts in either the non-responder or the non-responder. We're going to look directly at the upstream regulator. So which transcriptional program can be set? So everything you see here, the orange color and the blue color are the prediction. And the prediction is possible because behind any relationship of a genes downstream of a particular regulator here, catenin or any others, there is findings. And these findings are contextual and directional. So we can infer activation, we can infer downregulation, we can infer activity downstream and upstream. It's really powerful. As I mentioned this morning, this is typical. Even if you didn't have IPA, we say, oh, this is a signature of EMT. But let's be unbiased. Let's find out. If you were pulling all these transcription regulators predicted to be activated with the transcription regulator predicted to be inhibited, you effectively have an epithelial to mesenterial transition processes that show up to you. And this process is up. So this is a typical program. I'm going to select only 2 predicted to be activated, 1 predicted to be inhibited using that simple network, one-on-one relation. I showed that this morning. And this is one of the simplest hypotheses. 2 of these transcription factors that you see downstream or in orange are predicted to be activated. And all of that is based on the pattern of expressions that are known to be downstream or upstream of these guys, downstream of these guys. One factor, the SPDF, extremely important, is in general predicted to be turned down. It's downregulated in many metastatic processes. And these guys allow the cells to migrate, to be invasive. So this is the simple hypothesis you can test. We have an empty signature based on 3 factors, 2 predicted to be up, 1 predicted to be down. That's very cool, right? Well, we can go a little further. We are going to be very stringent in the way we construct our regulator, and that it's called a causal network. Causal networks are essentially a network composed of a master regulator and the 3 purple are the regulator together. And these guys are connected uniquely by activation and inhibition edges. So we can really mention if one increase, one decrease, we can infer that type of status. And that network of regulators sit on the top of target downstream. Each of these regulators has to be connected to 4 target downstream, and they are connected uniquely by expression and transcription edges. In other words, now we can really determine a direction, right? The beauty of that causal network is now I can ask another question. I can ask the question is, well, it's very interesting to have that master regulator connected with all these downstream target genes, but what it has to do with a specific biological processes of interest. So we call that a notation or scoring. We're going to use a master regulator that drives the causal network associated with a particular process of interest. Obviously, this is a process of interest that you would like to inhibit or revert. This is what I've done. So I look at the causal network analysis under the tab upstream analysis. You have 2 tabs, upstream regulator and causal network. And in the causal network, I say, well, now show me molecules that are predicted to be inhibited. And they are predicted to be inhibited because these guys here, the target here, are known to be downstream of these particular genes here. One of them, I select one of them, it's called a PEPB1. So even if you didn't know PEPB1, you can go in the search in IPA and you're going to have a gene view, and the gene view will describe you everything about that gene. What is the role, what is the pathways, is there any type of other connection with other genes, the disease, is it a biomarker, and so on. Well, these guys have been involved in numerous human cancers, and it may act as a metastatic suppressor genes. So it's interesting that IPA, based on the pattern of expression of genes downstream, predict that that guy, PEP1, is predicted to be inhibited. For the sake of the isoform view, I show you two isoforms differentially expressed, unfortunately not significant in that case, but we can represent them. If they were finding, this is new data, we could find more information about that. But let's do that network. So this is the network. This is PEPB1 predicted to be inhibited. It's in blue. And remember, I talked to you about annotating or scoring biological processes. Well, we're in advanced melanoma patients, so I wanted to bring that advanced melanoma function here. When that causal network, driven by a predicted inhibited molecule, in fact, which is called RAF kinase inhibitor protein, predicted to inhibit it, that drives the advanced melanoma up. It's increased, it's a prediction. And all of that sits on the top of target downstream with their upregulation and downregulation. You recognize many of them, SNE2 and so on. Well, the goal of that is to mimic an inhibition. So I would like to inhibit. So I'm showing that step right now, but the idea was, okay, how can we increase the activity of a particular molecule? It's pretty difficult, right? Well, you go in the knowledge base, and you go in tools that are called grow functions. And I ask the question, say, grow any type of molecule that is indirectly or directly associated with PEP1 and connect it. And I would like to see what's the effect. Well, for most of you, you recognize this is an anti-CD20 monoclonal antibody. This is not for melanoma. It's for another type of disease. But one of the indirect effects, and you find that by clicking on that edge here, one of the indirect effects of rituximab is to promote the upregulation of PP1. So why not? It could be a nice hypothesis to test in the lab. And I can back that up by papers in 2012 showing that effectively if you target a subpopulation of cells that a priori that has nothing to do with melanoma, like the B cell population, you may be able to revert some aspect of the metastatic. So that could be tested in the lab. Now let's go to the IsoProfiler. An IsoProfiler is the idea of selecting across all your thousands of splicing variants that have been differentially expressed in your data set to look some that are tied to a particular process that you would like to. In that data set, with a p-value over 5, so everything is significant, we have over 4,000 isoforms differentially expressed in the non-responder. And here you see the name, you see their type, the gene or disease function findings we can have, and the expression pattern. And you see the term GTEx, I will come back in 2 or 3 slides about it. And here, this is their expression pattern. And I can use different filters in IsoProfiler. I can say, well, I would like to have isoforms that are differentially expressed with a fold change cutoff of 1.5 absolute value. And I have a minimum of 10 count. And by the way, I would like this splicing variant to be of a protein coding biotypes. I don't want any other bad stuff. I want the protein coding because I can target that eventually. And I would like isoforms that have findings related to, so I can relate to something and I can read something about it. And I would like to tie that to, it's an autocomplete system, but I tap invas and it shows me all invasions. So I would like isoforms that are 1.5 cutoff, 10 RPKM, protein coding with some functions that are tied to invasions. From 4,009 isoforms have pertinent function to invasions, any type of invasion across the findings. One of them is the VGFA. This is the VGFA, and the representation of all the splicing variants according to the ensemble version 86 model. One of these splicing variants, the VGF165, the protein coding transcript support level 1, so this is strong, and yet, according to APRIS, it's an alternative form. It's upregulated in the non-responder. So it's very interesting. And we do have findings, so let's find the findings. This is the isoform views, and this is the pertinent differentially expressed significant isoform called VGF01. The name is VGF165. And if you click on that, here you will find findings that are really pertinent to your data set. Two of them, that particular form has been shown to increase the growth of melanoma in skin mouse, and you have the paper. And if you doubt, you have the PubMed ID and go get the paper. And we have a mutant information about, not in this one, we have also information about the promotion of angiogenesis of that particular form in skin mouse as well. So you have an idea now across all your 4,000 isoforms, you have one tied to invasiveness. And I think it's nice to pursue that idea and to check if it's interesting. Now let's go to the GTEx. OmicSoft has processed GTEx entities. And the power of OmicSoft is they are able also to process that at the transcript level. So the one-on-one connection was easy. Now we have IsoProfiler. We can look at isoforms that are differentially expressed. Let's find isoforms that are differentially expressed but are also enriched in tissues using that GTEx power. And obviously with GTEx, you can answer all these type of questions, right? Either any form enriched in the livers in my data set, either any differentially expressed isoform specifically expressed in a particular tissue. All these kind of answer you can imagine. I show you here the description of how OmicSoft has computed the FPKM at the transcript level of all these different files. More than 9,000 samples have been processed. We have excluded only 88. So we have 9,000 samples flagged by GTEx. We have a median FPKM, and they have calculated an enrichment score, right? So we have a value in IPA when the enrichment score is superior to 5. We consider it's enriched in the tissues. This is an example here in the adrenal gland. You don't see, but it's red. Here you have a form which is highly enriched on adrenal gland. So let's apply that to our data set. And I'm going to use some filters as well. I'm going to say for change of 2, RPKM count of 20, protein coding, isoforms, information about GTEx tissue expression enrichment. This is the filter. And here I can say, well, I would like to have isoforms that are enriched in the skin, in the liver or the lung. I selected these tissues because obviously they are the site of metastasis in melanoma. And I launch and I apply. And now you have 154 isoforms that are enriched in either of these tissues. And now you can scan. I selected DAC2. DAC2 seems to be downregulated here. Let's find out. So this is the profile. When you click on DAC2 line here, you are going to have these, the schematic of the structure, the name of the protein, the name of the transcript, and here you have in yellow basically the score of the enrichment in specific tissue. This is the skin tissue. Trust me, it's skin here, right? And these particular isoforms are effectively downregulated. So this is enriched in normal skin tissues. And yet, in our data, in a non-responder, it's downregulated. Well, it happens that that form, when it's upregulated, decreases the proliferation of lung cancer in certain settings. Well, it's downregulated, so there's a match. And here you have mutant information that has been observed in specific adenocarcinoma. So you have very relevant information about specific isoforms that are enriched in tissue because we have isoform and GTEx information from OmicSoft. That's one thing. So you can imagine doing a track on that particular isoform. So I show you who are the isoforms here. This is the two enrichment things. It's enriched in many other tissue, and it's interesting because it's enriched also in the track. The DAC2-001 isoform, it's enriched in the brain tissue. Brain is as well a distal organ for metastasis in melanoma. So now let's go to the big part, which we really like. I talk about the analysis match. And what we want to do, see, this is a screenshot of what we have in analysis, right? We have upstream regulator, we have causal network, and we have these different columns here. And you can do that multiple times, and you are looking for canonical pathways, upstream regulator, causal network, disease and function. All of that is fine within one data set, right? Before you could do, in IPA, you can do 20 observations of that, different time in one data set. It's cool. But let's imagine now that you don't want to think about it and you want what we call an automatic discovery across the land of data sets that have been pre-computed. And that's what happened. Now we are going to have a tab, and I will show you the tab, that automatically match against your own analysis in your project, but across 6,000 expression data sets that have been processed and curated by OmicSoft. It's extremely powerful. And you can do all sorts of things, obviously, right? Build your confidence in your analysis results, looking for anti-match that give you new insight, and so on. I like this slide because it shows you the together better. That will be the motto, together better. OmicSoft have processed and curated thousands of data sets, from Geo, TCGA, SRA, AeroExpress. Ingenuity system has provided thousands of peer-reviewed articles or findings from clinical trial and other database. And the two together, they merge. And now we are going to be able to analyze the biology at a higher level, at a stronger level, at a better level. And we are going to compare these analyses to all OmicSoft pre-computed data set. This is exactly the tab you are going to have. See, it's called Analysis Match now. When you get that, you will be very happy. So it's organized as almost like an Excel spreadsheet. You have, and that may change. Remember, we are the 19th of September. In September 30, we are still tuning that, but we are going to have the name of your analysis. This is the analysis you are going, what we call a query analysis, transcriptome, melanoma, non-responder versus responder, and I'm going to look through thousands of data sets, right? This is the project in which the data set came from. Could be mouse disease, human disease, OncoGeo, and so on. And we have many columns that we can set up and customize here. Oh, it's not there, but that will allow you to give parameter of interest. I just show you things that really interest me right away. I want to know what type of comparison you get, treatment versus none, disease 1 versus disease 2. And here you have the contrast, if there any time, if there any product you had in that data set, the type of tissue in which the data set has that expression profile, is there any disease that you can look at specifically. All of that are using a filter, so you can search. And here there is 4 columns. I only put 5 columns. And you will see purple, pink, purple, or blue. The purple indicates like a strong match, and the blue, a strong anti-match. And here it says, we're looking at the match for canonical pathways, upstream regulators, disease and functions, causal network, and we have a general z-score, an overall z-score, or general match. Does it match strongly or not? So you recognize that data. So what do we have now in IPA? Store and analyze in IPA. Remember, I need to mention that all the data set that has been pre-computed from OmicSoft now is in your libraries, in your project managers, are read-only. You can open them, look at them, you cannot modify, you can't copy them. That's done on purpose. And we have all these 6,000 data sets from disease land, human and mouse, from OncoLand, OncoGeo, TCGA, hematology. It's really powerful. So now my own data set on melanoma, I'm going to scan against 6,000 data sets. The lands, you know that, they represent different disease, different tissue, different platform, many things, different cancers. And this is based on comparison, right? So this is all the comparison we can get from that integrations. So again, I'm not a bioinformatician, but the strategy they have used to create the obtention of a query analysis. So in all OmicSoft datasets, you have upregulated and downregulated genes. They are comparison, right? What has been done first, they have filtered that by a p-value 0.01, just to cut the number of genes that are significant. If it's below 50, you trash. If it's not, you're going to keep that. And you are trying to keep a balance of roughly 1,000 genes if you can. And you are going to keep the balance of the genes that are up or down regulated. So one example. In that GSE 3188, which is a breast cancer test one, you can check in Disease Land, there was 54,000 prop set. 1,400 were upregulated, 651 were downregulated. We have 2,000 genes. We pick up only 1,000, and we maintain the balance. So we're going to keep for the query analysis only 700 genes upregulated, 290 downregulated. And now, this is the representation. In OncoGeo, this is the data set we want. We're going to keep these significant downregulated, these significant upregulated for the query analysis. Now we have the query analysis. Remember, IPA calculate the predicted to be activated or decreased for canonical pathways, for upstream regulators, for disease and function, you name it. Well, we are going to select the top 50 most and bottom for the causal network, upstream regulator, and disease and function. Z-score, the Z-score cutoff is absolute value of 2, and the p-value is 0.05. So we are taking the top 50, bottom 50 that have a Z-score of absolute value superior to 2 and p-value over 5, right? For pathways, is a little harder, so we keep only the 10 most and the 10 bottom. So now we have a kind of a query signature. We have roughly 100 genes, if we can, 50 up, predicted to be up, 50 predicted to be down. And now we're going to look at all these analyses that also have been analyzed in IPA, these 6,000 data sets. And all of them for upstream regulator, for causal network, disease and functions, and CPs, they have also a signature, right? And the goal now is to match. There's these 50 top are represented in that other data set. These 50 bottom are represented in this other data set as downregulated as well. If that is correct, it's called a strong match. So higher score. You can imagine 100% is the perfect match. In other words, your data set against your data set should be a perfect match. And you can imagine when the top predicted activated are found inversely here, and the top decrease are found up here, it's an anti-match. It's very strong anti-match. And you have all the category intermediate, right? So now you have an idea, okay, I'm going to be matching against other data set or anti-matching. So I passed the calculation of the Z-score and the p-value, but roughly you have a percentage. 0 to 100 for the match, 0 to minus 100 for the anti-match. And we assume a strong p-value that the calculation is there. This is the representation of our melanoma transcript level against pre-computed data set and your own, right? All of there. First things I'm going to do, I'm going to filter. It's too much. There's a lot of data. It's over 6,000. I'm going to filter in the project, look at analysis in only OncoGeo. Look only for disease versus normal. Remember, we are in a disease setting. I would like to look at disease versus normal. I will consider that responder is the normal and the non-responder is the disease. And in the contrast, I will put all terms like our prime for primary, met for metastatic, and cancer. Like that, I gather as much as I can. By doing that, you have these type of data set, 15 of them, only 15. The top here, so you see it's all pink and all blue. So the top pink, I would consider they have a similar pattern in the transcriptional and biological process signature. So they mimic. 8 in the bottom have an opposite pattern, anti-match. So now you have your melanoma dataset at transcript level, and you are going to find out that among all your criteria with all your filtering structure, you have 8 or so that dataset across other tissues, across other type of disease that match, and 8 or so that not match. And we are going to send that to a heat map. This is a heat map. In red here, we color them in red. This is your data set. So remember, this is the data set with 100 score, right? We take the top 50 and the bottom 50. So we have always 40 square like that going down, right? And this is the other data set. This is so powerful, and now we can click. Just to show you an example, we have imported also the metadata that exists in OmicSoft. And this is all of that. So you can scan and search. Here, that particular example is a skin melanoma. And it's an AFI platform, and so on. You have many informations. And you have a filter system here and here. And I'm going to use that because there is a lot of match. The match are the causal network, CN, UR for upstream regulator, DE for disease and function, and so on. So it's kind of a foggy right now, but I need to see what's going on. You can see that your data set is blue here, so it's predicted to be inhibited for all these components. It matches very well with these guys, and it clusters very well with these guys. It's very good news. We can cluster that. And I'm going to filter on the upstream regulator and on the Z-score. And on the upstream regulator, I will filter on the molecule type. Look for only cytokines that are upstream regulator and growth factor that are upstream regulator. And I'm going to put a Z-score of 4. I want something really of high-level predictions. And now we have only that, 15. And you see again, this is your data set, cluster very well with these guys. Everything you see here, these upstream regulators are predicted to be increased in this data set. And all these upstream regulators are predicted to be decreased in this data set. And you can see there's 2 strong clusters here. It's very interesting, right? So just for the sake of the example today, I'm going to say, well, it's very good. All of them are cytokines and growth factors. This is them. Let's add them to pathways. Simple. And remember, I'm going to take that example. They are all predicted to be increased. And I put them to pathways. They are all predicted to be increased. I use the map molecule activity predictor, and I mimic them by painting them in red, which means predicted to be increased. And I ask the question, well, go in our knowledge base and find functions that are tied to these guys. So each of these guys are tied to these 3 functions, and each of these functions are tied to the 15 genes. This is what I call an upstream regulator signature related to tumor progression. As you can see, 3 functions: migration of cells, development of new vessels, and cell proliferation. They could be used as a marker of tumor progression. That's one example. And you can look at these guys and mimic and overlay the data set of your own, the expression of your own data set or your own predictions. That's one example how we can use analysis match. It's only one of them. There's 100 of them, but that's very powerful. 4 click. Now let's go back and let's abandon analysis match, but let's go back to OmicSoft Land. Remember, I said that we can look at upstream regulator and I can select growth factors. And I would like growth factors that are predicted to be activated in the melanoma non-responder patients. One of them is the FGF2. I know, sounds familiar, we have talked about that for 3 talks, but this is one of them that we can look at. So FGF2, even though it's slightly transcriptionally downregulated, it's functionally being boosted here, and it's based on the 61 gene and their pattern of expression. We can predict that FGF2 has been activated. This is the network of interest. This is the isoform we talked about this morning. And now I go back to OncoLand. So remember, 6 months ago, I could not understand how OmicSoft work and OncoLand works. And now we can do that. I can do that. I'm going to understand the FGF2 expression and the relation with melanoma and other point of interest. And here, this is the FGF2 expression according to different tumor types. You all know that. Skin melanoma is there. There's a lot of sample, very crowded. I need to clean that. And I'm going to use a LAN query. And I'm going to say, well, I'm going to look at the RNA-seq FPKM level of FGF2 and establish quarters, low expression, low medium expression, medium high expression, and high expressions, right? And query that on the metastatic melanoma skin samples, uniquely metastatic melanoma samples. And this is what we do in 4 clicks. I have all these metastatic skin melanoma samples according to the low or high expression of FGF2. So I'm going to look only at the low expressor of FGF2 and the high expressor of FGF2 in melanoma metastatic sample. Joe showed you that this morning. We have used that here as well. We make another query, and we have the question, basically, I'm interested in, okay, I know that BRAF is important melanoma. Can we find any type of association? And it's really not because I wanted. It's because the tools show us, you look at BRAF is the top 5 genes strongly associated with my query of interest, which means low expressor of FGF2 and high expressor of FGF2. Boom. So now, if I didn't know, I check BRAF. Well, show me BRAF in the OncoLand and show me in a sample. And you recognize this is the skin melanoma sample. In more than 50% of the sample, BRAF is mutated in melanoma. Good news. Let's go further. We can look at the expression of FGF2 based on the fact that BRAF is mutated or wild type. And this is a box plot. You can tell in the mutated sample where BRAF is mutated, the expression is higher of FGF2 versus the wild type. Well, there's not only one mutation of BRAF. There is multiple mutation of BRAF. So we can query that in OmicSoft Lands, in OncoLand. And this is what we have done here. I asked the question, could you spread and represent to me, based on the expression of FGF2, based on the presence of the mutant and the different mutation of BRAF, what's going on at that correlation level, FGF2 expression, BRAF mutant? Well, you can see quickly, right? This is the V600E. The patient in that paper, the vast majority have a V600E mutations, right? You can tell quickly that higher the expression of FGF2 exists in the sample of metastatic melanoma where the V600E mutant here. This is the wild type here. So what I wanted to show you is, even for a non-expert in bioinformatics, I can do a lot of things by hand just by clicking in all these fantastic tools. And this is what I found here. So in conclusion, now I can analyze data in RStudio with the help of Joe, but it's getting there. It's getting easy. You send the data to IPA and you can answer all these questions. You can look at significant gene and pathways. You can identify specific enriched splicing variants, so molecules that could be of interest. You can generate novel networks that you can test and inhibit in the lab. You can compare that analysis in OmicSoft Lands using analysis match. Really powerful. In 4 clicks, I could say, well, we can find a cytokine, growth factor, upstream regulator, signature. You can test in all your data set now. And you can visualize that FGF2 of interest in the whole scheme of OncoLand and other genes. And we have found that FGF2 BRAF correlation. So yeah, it has been a great journey using that tool. So thank you very much. So a big thank you, just to finalize, to the Bioinformatics teams. And we are around the world, as Frank has said, North Carolina, Cary, California, and Romania. And specifically, I would like to thank these people, Joseph, Stuart, Jeff, Andreas, Jens, and Gabriela. All great minds. So thank you, guys.
Now that the analysis manager is implemented, and my understanding is this morning there was quite a bit of work done to improve the ontologies that were used behind the land samples. So I was wondering if there's an opportunity to combine those. For example, could you do an analysis match and then look for ontology terms that might be enriched or overrepresented
So, well, all these 6,000 data sets are carried over, automatically analyzed in IPA, and they carry the metadata. So that's the thing you can look at. Each of these analyses can be open. It's a read-only. You can open, and you can see what is enriched in function and upstream regulator. You can't modify, you can't copy, you can't do anything, but you can see that, right? So that's the only way we do. Remember, this is the first ever versions. As of now, we are working on boosting that in a strong way. We have so many ideas to do. It's the first shot, right? So yes, we carry the metadata. The metadata could be searched in the future. We can see, we can open any analysis. That's okay, depending on the license, and it's really really powerful. That answer your question? Oh, it's literally unbelievable. In 5 minutes, remember, I'm like a customer inside. I'm paid to play for that, right? And it needs to tie to something concrete that I can relate to the collaborator that we are really publishing. This is not a joke, right? And this is unbelievable. Now, one click. It's when you open your core analysis, the analysis match, it's already done. It's done. There's nothing to wait. It's coming. And now you can click. And in a couple of clicks, you can select based on processes. If I wanted to look at disease effect analysis, these guys, I could have selected here only the biological processes. Maybe I would have found match of completely antagonist process carried by the same transcriptional program. You imagine, you can look at one specific transcription factor across all the analyses. I would like to see what MIC is doing across all of these. You can do that. Just by a matter of filtering and clicking, you can get that. I was blown when I tested that the first time. I said, wow, that's pretty cool. That's it? Cool?